Hi there. So now that we have reminded ourselves of how nervous system can be divided into central and peripheral nervous system, it is a good time for us to start looking in more detail into the central nervous system. And I want to start with this next thought. If we were to look at it brutally, simply from the point of view of different tissue types, the brain is nothing but a lump of fatty tissue, right? But then again, it is within the brain where you really exist. It is there where your personality and memories and dreams and imagination, as well as wishes and future, and all other things that make you the you that you are, they take place in your brain. It is your brain that makes you the unique and fantastic individual that you are. So I guess that it could be said that the heart has nothing on your brain. Maybe it is pictures of the brain that we should be drawing and giving to each other on a Valentine's Day note. Well, maybe not. So I guess that our first question should be, how did we end up here? And how do we compare to other species? What we see here in color is a tree-like diagram, phylogenetic tree, which shows us relationships of some mammals. And as we are starting to look at it, let's remember that nearly all animals have some sort of nervous system, right? Here you can see various organisms and how their nervous system's level of complexity varies. And what is kind of cool is that if we focus on looking at the brain, on these examples of our phylogenetic tree, we see that no matter how different these species are, they all have similar regions, right? Well, we will be focusing on the human and sheep brain. And this is because we will be, of course, studying the human body, and then we will be using the sheep brain to do our dissections, while comparing it to that of a human brain. Well, the other thing that I wanted to talk about with you is that when we look at the evolution of the skull, we notice something interesting. Over the past 3 million years, the size of a skull grew from an early human to the modern human. And this growth was really associated and likely driven by the increase in the size of the brain. And one thing that we notice especially is that the size of the anterior portion, this is our frontal lobe, grew especially during this evolution. And another thing that we see really obviously is the folds that appeared. This is to facilitate a greater surface area. Just like when you are crumpling a paper, you can pack a large surface area in a smaller space. So that is what we did for our brain over the evolution. So both of these characteristics, a large frontal lobe and a great amount of surface area, must be really important for us as a human, a modern human. Well, the next thing that I want to discuss with you is that what happens to the central nervous system, and in particular the brain, during our development, over one individual's lifetime. Let's have a look at that. So brain is really one of the first systems to start developing. Although at the early stage it is not terribly well developed, it is still there. And it grows on a fast rate during prenatal development, so this is before the birth, and also during individuals' early life. So note how compared to many other organs that are finished by the time of the birth, the brain still keeps developing past this point. And then as you follow the diagram further, the rate of development slows down, but it never stops. And what we see is first just this neural tube, which then thickens, folds, and specific regions appear. Let's have a look of that. So here we see first is just a stick-like neural tube that starts to take shape and different areas of it develop, and these different areas later give rise to the different parts of the brain that we will see. And one thing that we will see over these next three figures is that as the brain grows at a faster rate than the rest of the skull, it must fold to fit into the space available. And this is how we get this shape and especially the folds of an adult brain. And I want us to look at this a little more in detail. The four regions of an adult brain are the cerebral hemispheres. These are the big parts that we see on the outside. Really what most of us think of when we talk about the brain. Then we have the diencephalon, which is the parts buried inside and in the middle of the brain. And then we have the brainstem. This includes three main parts, the midbrain, pons, and the medulla oblongata, often simply known as medulla. In fact, what is interesting about the brainstem is that it is the first part of the brain to develop and houses some of the vital functions like breathing and heart rate centers. So things that we really need. And in terms of being the first to develop, I mean that it comes about as one of the very first parts of an individual, but also came about first in evolution. So it is very important, and therefore pretty well protected in its location, right? And finally, I want to include the cerebellum, which is literally the little brain at the back. We will go through all these in a bit, but first I wanted to introduce you to the concept of gray and white matter. I don't know if we have with us anyone else who watches or reads Sherlock Holmes stories. These stories created by Agatha Christie. Sherlock often says that he needs to dig into the gray matter, right? So what does this mean? Well, it refers to the fact that he has to think about whatever the crime mystery he is solving. So this suggests us that the thinking would be happening rather in the gray matter than in the white matter. What we see here is a cross-section of the cerebrum, showing how the outside of the brain, mostly, this is our cortex, have the gray matter. And on the inside we have white matter. Well, to understand what the gray and white matter are, we must go back to what we learned about the neuron structure in our earlier chapter. Remember the cell bodies and myelinated axons. So it was myelinated axons that served the passage of a nerve impulse mainly. So these would show up in the nervous tissue as paler areas. And then again, darker areas do not have myelin. They are mainly cell bodies and so on. So what we see is that the center of the brain is made of myelinated axons and is whiter in color. So to simplify a bit, it is areas where messages are just mainly passed on. And then cortex, the outermost layer of the brain, is darker. And this is because it contains more nerve cell bodies. And this is where the processing of nerve impulses takes more of a place. So we are starting to see that anatomy gives us a cue about the physiology, right? And we will see the same pattern when we dissect a sheep brain. And what is kind of confusing is that this is actually reverse in spinal cord in terms of the colors and their locations. Kind of cool, huh? The other thing that we can see a hint of here is that inside the brain there are all these empty spaces. Let's look at them a little more. And what we see here is the ventricles of the brain. These are fluid-filled empty spaces inside the brain. And interestingly, they are all connected. And the fluid that flows here is the cerebrospinal fluid, or CSF for short. And if you think back on our previous chapter on different glial cell types, these were lined by the epidermal cells, right? Well, this concludes our introduction to the central nervous system and brain in particular, and we have also talked about the evolution and development of an individual's brain, and then we even managed to do a brief introduction to the 4 main parts of the brain. A good start, I would say. Let's continue in the next video.